Okay, in today's class, we're going to discuss about the difference between chromosome, chromatin, DNA, and gene. Let's start with the chromosome. Here we have the chromosome, the X-shaped structure. You all are familiar with this one. If we zoom into the chromosome, we will see this thing. This is called chromatin. In other words, I can say that chromosome is made up of chromatin. If I take this chromatin, okay, and if I'm going to wind it up into X shape, what I will get? I will get the chromosome, obviously. See, if I get the chromatin is this type of thing, okay? If I'm gonna wind it up into an X shape, I will end up with chromosome, right? Now let's zoom into this thing. What is it? If you're gonna zoom into this one, we will observe that it looks something like this. We will see that the chromatin is actually made up of two things. It is made up of this blue colored thread and this coin shaped thing, right? We can spot only two things. We can see the blue colored thread and we can see the coin. What are these two things? The blue colored thread is basically the DNA. This one, this blue colored thread, okay? This is the DNA, and this coin shaped thing is called protein. This is a protein. And you can guess the name of the protein. It's histone protein. So I can say that chromatin is a combination of DNA plus protein, right? Here we have the chromatin. And chromatin is a combination of DNA plus protein, okay? So, in other words, I would say that in a chromatin, a DNA is wrapped around histone protein, okay? This whole thing is the chromatin. Let's zoom into this one, okay? You know, this single thing, okay? I'm talking about only this thing. This single thing is called nucleosome. What is nucleosome now? Nucleosome basically means when a small section of DNA wrapped around histone protein is called nucleosome. Nucleosome is this one single structure, okay? This is called nucleosome. When I'm going to attach many nucleosomes together, what I will get? Chromatin. Let's get back here, okay? I'm going to summarize this one now. What is chromosome? Chromosome is X-shaped structure. We all know about chromosome, okay? When we are going to zoom into the chromosome, what we will see? We will see the chromatin. This is a chromatin. It looks like a simple wire type thing, but it's not that simple, okay? If we zoom into chromatin more, we will observe that chromatin is actually made up of DNA plus protein. Okay, this DNA and all of these proteins together is called chromatin, okay? And this single thing, this one, all of them together will be the chromatin, but this single thing is called nucleosome. Nucleosome is a combination of DNA plus one histone protein. One histone protein and a small section of DNA would be the nucleosome. All right. Here we can see that this is the nucleosome. You know what's the other name of chromatin? The other name of chromatin is polynucleosome. Many nucleosomes together is called chromatin. Okay, anyhow, here we have the blue colored thread, which is the DNA. Here we have the DNA. That's how it looked like, right? What we're going to do is we're going to dive deeper into the structure of the DNA. We all know that DNA is double helical structure. It has two strands, which are in spiral shape, and they're coiled around each other like this, right? This is the simple DNA, obviously. Let's zoom into this one. If we're going to zoom into this one, okay, you know what we're going to see? We will see these things. Let me make it a bigger so we can see better. Just a sec. I'm gonna draw it more big. Okay, now let's zoom into here. Okay, here we have the one strand, here we have the second strand. Let's zoom into this strand. You know what I will see here? I will see a sugar. You can guess the name of that sugar. Deoxyribose. Yeah, it's this deoxyribose. I'm drawing deoxyribose sugar right now. Yeah, here we go, the deoxyribose sugar. We should have 5 carbons here, right? Carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. Let me write 4 clearly. Here we have the 4. And the carbon number 5 is attached to the carbon number 4, which is here. The green colored thing is a sugar, deoxyribose sugar. Okay. Now, A base is attached to the carbon number 1. There are 4 types of bases. There are 4 bases that are present in DNA. We have adenine, thymine, cytosine, and guanine. We have these 4 bases that are present in DNA. Alright, so what can happen is I will attach one of these bases here with the carbon number 1. Let's attach cytosine here. I will take C and I'm going to attach with the carbon number 1. Okay. Now with the carbon number 5, a phosphate group is attached to the carbon number 5. That's it. What we have done, we have taken a sugar, okay, and we have attached base with the carbon number 1. We have taken phosphate group and we have attached the phosphate group with the carbon number 5. The combination of these 3 things is called nucleotide. It's nucleotide. Don't mix this nucleotide with the nucleosome. This is a nucleosome. This is a nucleotide. Both are completely different things. Okay, yeah, here we got the nucleotide. You know what a DNA is? We call DNA as polynucleotide. DNA is a polynucleotide. What does it mean by polynucleotide? You might be familiar with the word poly. Poly means many. Nucleotide means this thing. DNA is a polynucleotide. That means DNA is made up of many nucleotides that are connected together. I have just drawn one nucleotide. Let's make a second one. I'm going to make the second nucleotide. Let's do that. I will take the sugar. I will draw the sugar here. Here we have the oxygen. Here we have carbon number 1, carbon number 2, carbon number 3, carbon number 4, and here we have the carbon number 5. This is the sugar. What I would do, I would attach any base with this one. Let's attach adenine here. With the carbon number 5, I'm going to attach phosphate. It's the universal rule. Adenine, any base will attach to the carbon number 1, phosphate is going to attach to carbon number 5, okay? Okay, here we have 2 nucleotides, 1 and the second. I have to attach these 2 nucleotides together. How? What I'm going to do is, I will create a bond between this phosphate and the carbon number 3. This is the bond. What is the name of this bond? This is called phosphodiester bond. This yellow bond that I've created between the carbon number 3 of 1 nucleotide with the phosphate of second nucleotide, this is called phosphodiester bond. I have connected 2 nucleotides together. Let's draw the third one. To make it more simple and easy for you, guys, what I'm going to do is I will just draw DNA in a straight line, so you can guess better. So okay, let's do this way then. Yeah, okay. Here we have the one strand of DNA, and here we are gonna get the second strand. Okay, this is just for the sake of understanding. I'm just gonna draw a straight line rather than the spiral one. Okay, so we have drawn 2 nucleotides. I'm gonna draw the third one now with the same way. Carbon 1, carbon 2, carbon 3, carbon 4, and here we will get the carbon 5. I will attach the base with this one. This time, let's attach guanine. And with the carbon number 5, I have to attach phosphate, and I need to connect these 2 nucleotides together with the phosphodiester bond between the phosphate and the carbon number 3. That's how. This is what you're gonna observe when you're gonna zoom into one strand of DNA. You will observe like this. Okay, we have drawn the one strand. Let's look onto the second strand. What can happen here onto the second strand? Second strand is also made up of polynucleotide, obviously, but there is a single twist. The twist is this sugar get inverted, okay? The second strand has inverted sugar. And what does it mean by inverted? Inverted means upside down, okay? Upside down means like this is sugar. Upside down means it would be like this. This is upside down, obviously. So yeah, let's do that. Upside down means the oxygen would be here and here, 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 and here. Here we're gonna have carbon 1. Let me draw with the other color. So okay, I'll take orange color again. Carbon 1, carbon 2, carbon 3, carbon 4, and here we have the carbon 5. The sugar is inverted. The other rules will stay the same. We have attached base with the carbon number 1, right? I'm gonna do the same thing here. I will attach base to this carbon number 1, okay? So I will attach the base with this one. And I will attach the phosphate with the carbon number 5, right? The thing that we did here. I'll attach the phosphate here. What we got? We got one nucleotide. What I will do now? I will draw the second one with the inverted sugar, obviously, because here we have the second strand. So I will draw the inverted sugar this way, this way, this way, this way, this way. Here we have the oxygen. And let's draw the carbon number 1, carbon number 2, carbon number 3, carbon number 4. Here we put the carbon number 5. Yeah. Now let's draw the base here. This is T, okay, this is thymine. T here. And I will attach the phosphate here. Yeah, and what I will do is, what I did there, phosphodiester bond. Same thing here. Phosphodiester bond is present between carbon number 3 and the phosphate group. I will do the same thing. Carbon number 3, phosphate group. I will create a phosphodiester bond here as well. Okay. So that's what the DNA structure look like when you zoom into that. All right. You must be aware of the base pairing. Okay. The base pairing goes like these bases gonna combine. They're gonna combine together with the help of hydrogen bond. Okay, there is a hydrogen bond that is present between adenine and thymine. There is a hydrogen bond that is present between guanine and cytosine. Okay, these two bases are connected with the help of hydrogen bonds. Okay, A always pair with T, G always pair with C. Okay, okay. Now what I'm gonna do is the thing that we always study in school is we always say that the structure of the DNA is like the DNA is made up of two strands. One strand goes from 3' to 5', and the second strand goes from 5' to 3'. The DNA strands are anti-parallel. What does it mean by that? What does it mean by 3', 5', 3'? What is it? Let's see that now. Okay. Can you see here this one? The phosphodiester bond is present between the carbon number 3 and the carbon number 5. That means this is linked to carbon number 3 and carbon number 5. Okay, the name is according to these carbons. Let's see how. Here we have the carbon number 5, right? This strands of DNA starts with carbon number 5. Starts with carbon number 5. In other words, I would say that, here the carbon number 5 is free. C5 is free right now. Free right now means it's not attached to any other nucleotide here. There's no nucleotide on the top of this one. Okay, C5 is on the top right now. Look here. It ends with C3. So the name of this strand would be 5', 3'. Let's look at the name of this strand. Here we have carbon 3 at the top. So this is 3', and it's going to end with this one, this carbon 5. So this is 5'. So the two strands of DNA, 5', 3', 3', 5'. Both are anti-parallel strands. I hope it makes sense now. I hope you're aware now with the basic structure of the DNA and with the difference between the chromosome, chromatin, DNA. One thing left, that is gene. What is actually a gene? Gene is nothing special. It's just a small section of DNA. If I cut a small section of DNA, that would be the gene. Okay. And these small sections of DNA, okay, if I just cut the DNA into small pieces, that would be the gene. What does genes do? What does a gene do? It gives one specific feature. Okay. For example, let's suppose this is just for the sake of understanding. Okay. Let's suppose the name of this gene is gene A, gene B, gene C, gene D. The gene A will give, for example, you're going to give us a hair color. It will decide the hair color. And the gene B, for example, is going to decide the eye color. And the gene C can decide, for example, it will decide the height of the person. And the gene D, let's suppose, it's going to decide the skin color. So the DNA is made up of genes. Let's conclude the topic. Let's do that. Chromosome is made up of chromatin. Chromatin is actually polynucleosome, where many nucleosomes attached together, it's going to create the chromatin. What is a nucleosome? A nucleosome is the combination of one histone protein with a small section of DNA. What is a DNA? It's polynucleotide, where many nucleotides attached together, it's going to create polynucleotide, and the polynucleotide will be the DNA. And a small section of DNA is known as the gene. Yeah, that's the end of today's class. I hope now it's clear.